Hello and welcome. These are the interviews we do with the Department of Ophthalmology faculty that are really here to sort of let you get to know the people that are working here and all the science for vision restoration in the Department of Ophthalmology at the University of Pittsburgh. Today, we're interviewing Dr. Kun-Che Chang. Dr. Chang is an assistant professor of ophthalmology at the University of Pittsburgh School of Medicine. Kun-Che, very good to see you, and thank you for coming in and spending some time to do this today.
Thank you, Craig.
So you've been with the department for just about a year, if I recall. It's gone by pretty quickly, but and you're working in research and trying to help find ways to restore vision for particular types of vision loss. Mm-hmm. Before we get into what your science is, I want to talk to you about, why you got into such a direction in your life and in your field. Tell us a little bit about that history. Like what got you interested in science and what drove you to say, the area I want to concentrate and spend my life's work trying to help is people who have lost their vision. What are some of the things that led us to this point?
Sure. So 10 years ago, I came to US for my graduate study. And actually at the time, I studied the interior of the eyes, which is the lenses and the cornea. Okay. So I studied a bit like diabetic cataract and some cornea defect by hyperglycemic effect. So after graduate, I think it's time to study some of the posterior back of the eyes. Uh-huh. I see the photoreceptor and the retinal ganglion cells are two important neurons in the eyes and the retina. So I start look for my postdoc, and I was lucky to get the postdoc position at Stanford with Jeff Goldberg to study the retinal ganglion cell regeneration in glaucoma and in other optic neuropathies. So that's how I start to touch the retinal neuron in the eyes. Yeah, so back to like 5 years ago, I identified some intrinsic factor which is important for retinal ganglion cell differentiation. Uh-huh. And now I have my independent lab at Pittsburgh. So I want to apply this finding to the stem cell differentiation from the human stem cells. Yeah, so yeah, that's pretty much because we all know the retinal ganglion cell is a neuron in the eyes, which just kind of transfer the visual information from the eyes to the brain through its extending axon. And then unfortunately, retinal ganglion cell do not regenerate after it's dead or injured. So that's why I think it's important to find a way to find a new source of the retinal ganglion cell, especially for the patient. So I would say the human stem cell replacement therapy would be the feasible approach.
Wonderful. So people who would lose their vision in their retina, or from glaucoma, from macular degeneration, some of these things which affect the back of the eye, the area that you work, those conditions would affect the retinal ganglion cell, those nerves that deliver that communication from the eye to the brain?
Yes, there are many different neurons in the eyes, but particularly the retinal ganglion cell, or we call RGC, is extremely important because it's not just only interact with a neighboring neuron in the eye, but also it's a special function to transfer the visual information through its axon to the brain. Mm-hmm. So in some glaucoma patient, they lost their RGC, but they also lost their axon. That's why they cannot see. So photoreceptor is important, but I would say RGC is the main retinal cell to connect eyes to brain. Yeah.
And you said you're applying things like stem cell technology to try to regenerate, restore those retinal ganglion cells. So tell us about the stem cells, because I think some people don't quite understand where stem cells come from, how they're different. There's all types of different stem cells, from very, very immature stem cells to more mature stem cells to stem cells in different areas of the eye. how does it work with the stem cells that you're using for the purposes of your research?
Yeah, so basically there are about three main stem cells. So one is embryonic stem cells from the embryo, and one we call iPSC is an induced pluripotent stem cell. Right now, the scientists can derive this iPSC from the dermal of the patients, so they can use to study some genetic diseases. And one is called like a tumor cell over from the bone marrow. Also in my lab, I use the human embryonic stem cell to differentiate into the RGC. Yeah, because in our lab, we have CRISPR-9 engineered embryonic stem cell, which we can see RGC easily through is the fluorescent protein, which is the TD tomato.
Okay.
Yeah.
So there's another, thing I think would be interesting to learn a little bit more. You mentioned the CRISPR-Cas9 technology. So you're using that essentially and that's like a CRISPR-Cas9 is a gene therapy technology that's used to allow you to try to find a way to tell genes how to to maybe correct something in a gene or maybe change something in a gene, almost like a pair of scissors, right, that goes in and manipulates that gene. So how are you using CRISPR-Cas9 then to adapt it to essentially train those stem cells to do what you'd like them to do with the retinal ganglion? Am I getting that right? Or is it something?
Well, yeah, yes and no. You are right. The CRISPR-9 is engineered to modify the defect of gene, which cause some diseases in tissue or organ. But in my lab, I mentioned the CRISPR-9 cell line is the lab we incorporate with the lab from Johns Hopkins from the Danzak lab. So they created one cell line specifically express the TD tomato.
I see.
Drive by the BRN3B because BRN3B is the non-RGC marker. So we can utilize this cell line to easily visualize the RGC differentiation in the stem cells.
So it's very incredibly amazing and complex, but I think it's so essentially what you're doing is taking a cell line that's really already been genetically engineered.
Yes.
That we already know does work on trying that will work around the steps needed in the regeneration process with retinal ganglion cells.
Yeah.
Okay.
And this is the advantage of that because think about it, once we have the RGC differentiation, which express the color we call TD tomato, and then we can transplant into the host retina in the animal model, and we can easily to see the transplant cell or donor cell by seeing this color. Yeah, so that's why the purpose they're gonna generate this spatial cell line.
So what have you been able to see so far? I mean, have you seen some changes that are really getting you excited in growth of retinal ganglion cells maybe that have been damaged from glaucoma type pressure? Have you been able to see any restoration of those cells through your work?
Yeah, so talking about the glaucoma or RGC loss model in our lab, instead we use the retinal crush model instead of the intraocular pressure elevation method. Yeah, because in my lab, in cooperation with the TACA and other experts of the field in our department, so we used retinal crush to cause the RGC loss and degeneration. Yeah, so
you crush the nerve to essentially cause that same sort of damage that happens with glaucoma, because glaucoma essentially is pressuring the nerve and doing a crush on the nerve. And now when you do this in the mouse, you can then use in your case, your stem cell therapy. And in Takaku Ujima, who works closely with you, he's using the same line for different types of regenerative therapy. So I see.
Yeah, so in Dr. Kuwajima's lab, he focuses on the gene therapy. And in my lab, it's a similar model with retinal crush model. I focus on the stem cell transplantation. So back to the organoid, the stem cell. So we were able to differentiate the stem cell in a 3D culture, which we call retinal organoid, which means we can culture the stem cell in the floating tissue organ. So we can observe the retinal cup differentiation through the early stages. Around day 30, we can start to see the RGC-like cell, which express the TD tomato. And then for a couple months, around like 6 months, we will see the maximum RGC expression in the human stem cell derived retinal organoid. Yeah, and then through this retinal organoid, we can study many things. We can apply our finding in animal models to the retinal organoid model, which we hope we can improve more RGC differentiation in a stem cell, because like what I mentioned earlier, the stem cell transplant therapy will be the feasible approach to treat the RGC loss. So that's why we try to identify the new molecular pathway or a new strategy to differentiate more RGC-like cell from a human stem cell. And then our ultimate goal would be transplant this donor RGC cell into the host retina to treat RGC loss. Yeah.
So in the future, if everything goes well, what you're hoping to be able to do is, again, with this sort of line of stem cells that are sort of being developed, more or less manufactured, right? Yeah. You're taking those cells and then they would need to be surgically implanted, I'm imagining, right? Or maybe injected into the space, in there with the retinal ganglion cells. And then hopefully you'll see where you have damaged tissue, you'll start to see that regeneration. And that is that where we're going in the future, we hope.
Yeah.
Where if someone has unfortunately lost some sight because of damage to the retinal ganglion cells because of glaucoma, that we actually may be able to restore that. And that's the hope, that's the dream. Am I right?
Yeah, that's a dream. And our long-term goal in our department would be doing the non-human primate transplantation. Yeah, this is always the goal of our department. Yeah, although I'm doing the rodent model right now, but my lab, the long-term goal would be apply our stem cell therapy into the non-human primate. Yeah, it's our long-term goal before go to preclinical trial.
Wonderful. Well, it is very exciting, and it's certainly exciting that along with what you're doing, and we have work going on with gene therapy and other types of tissue engineering type of therapy, and we have all sorts of different ways that we're trying to address the same issue. And it's exciting that so much of this is happening here at the University of Pittsburgh. And you came, as you mentioned, from Jeff Goldberg's lab, Dr. Goldberg's at Stanford, and collaborated with many of our scientists and close colleagues. So I imagine your work, in addition to going on here at the University of Pittsburgh, you're collaborating with people all over the country, and imagine all over the world. So it really is exciting to hear about some of the things that are going on here, and by all means, we're really grateful that you're doing this here. I'm
happy to be here too.
Well, thank you, and we'll look for exciting things and look for updates from time to time on how your work is progressing. So
thank you,
Dr. Chang, thank you. Have a great day.
Thank you. It has been a pleasure. Thank you.